This video is for period 3. We're going to talk a little bit more about mutations. We're going to talk about a point mutation today, and hopefully you can see that. There's a little reflection here, and you can sort of see me. Point mutation is a gene in which one nucleotide in the gene is changed. So it's one of the bases. And I'll show you a picture on that in a few seconds here. When one nucleotide is added or removed, the effects can be drastic. And the reason why is because we are changing the amino acid that those letters code for. And so if we do that, it could have a terrible effect. So let's look at this sentence. I think the best way to look at a point mutation is to sort of see what happens if we just change one of the letters in the sentence. So the original sentence is the fat cat ran for the rat. And if we're going to do an addition, which means we're going to add a letter to this, you're going to see that since everything is read in threes, anything after that actual mutation is now going to be not making sense, as you can see, because we have the word the, and that was fine. And then before the word fat, we put a G in there. And by throwing the G in there, it now threw off the codons, and the rest of the sentence doesn't make sense. So anything after the mutation is going to get altered. If we look at a deletion, we have that same sentence, the fat cat ran for the rat. And now in this case, we got rid of the F. So the is still the same. We didn't change anything. But when we're now going to delete, we're going to get rid of the F in fat. And now nothing again is going to make sense after that, because it's going to be read as threes. So all triplet codes past the addition or the deletion are then going to be altered. And if we alter that, that's going to change the amino acid, because if you remember the three letters, if you have ACC, it codes for a specific amino acid. If you have AGG, it codes for a different amino acid. So that's where the problem could happen, where some of these effects could be drastic. This may result in the formation of a protein that is not functionally normal. And some mutations aren't harmful at all, so we're not saying that every time this happens that it could be a problem, but it's what is really happening here is it's a frame shift. Because we got rid of the F or we added a G, we have now changed the way that this is going to be read. So if we move on, this is just a picture of, again, a normal gene. And it is, if you look at the top picture, it created the messenger RNA strand. We've now made our polypeptide chain of amino acids. The amino acids are the ones that are in sort of the bluish boxes. And then if we go down to the bottom left, in this situation, we have done a substitution. So instead of I think it was ACC, we've now changed it to ATC, which means we've changed the messenger RNA strand. From that T, it would now be an A. And now we have SER instead of GLE, for going back to that original one. And because of that, we've changed that amino acid. It could be harmful. It might not be. And then if we look at B is an example of where we have a deletion. So now we've gotten rid of the A. So instead of AAC, there's only one A there. You can see the green A is sort of leaving. And now we have changed, again, anything after that mutation, all of the amino acids are in green now instead of blue. And those are the ones that have been changed. So anything after mutation is then going to get changed and shifted. And then one other thing I'm just going to turn over here that I wanted to show you was something that I can also link it up for you. This is just the difference between a gene mutation and a chromosomal. Chromosomal mutations are what we talked about last week. Gene mutations, if we deal with an insertion or an addition, it's when one base is inserted into the DNA sequence. Deletion is when one base is deleted into that DNA sequence, and what we mean by that is that base sequence. Substitution is one base is changed into another. The chromosomal mutations, they have duplication where you have extra copies of genes that are generated, or you add an extra copy. Deletion, some form of the chromosome breaks off and it's gone. We showed pictures of that last week. Inversion, broken chromosomes get reversed and put back into the chromosome. So they just get flipped around. And translocation is a fraction of one chromosome breaks off and attaches to a totally different sort of non-homologous chromosome. And we will talk about more of this when we do our next Google Meet.